are you?
Hi, Andrea. My name is Shu Lam, and I'm currently a PhD student at the University of Melbourne, Australia. So I'm also a leader of research in polymer and microbiology that we're working on. And we have recently developed a new class of antimicrobial agents that are capable of combating antibiotic resistant bacteria.
And why is this discovery so important?
I think that this discovery is really important because currently antimicrobial resistance is a really severe issue that is impacting healthcare on a global level. So for example, I think if I'm not wrong, in the United States itself, there are about 23,000 illnesses caused by these antibiotic-resistant bacteria each year alone. And a very scary statistics says that by the year 2050, 2050, if nothing is done to resolve this issue, we would see 10 million deaths caused by these drug-resistant bacteria each year, with one people dying in every 3 seconds. So I think it is a very serious issue, and currently we just don't have much available options to combat it. So I think that this research, this discovery that I've managed to accomplish, sort of sits in to address the gap, and it offers a way to potentially replace antibiotics and to effectively combat such bacterial infections.
Could you take us through the technical challenges you had to face? For example, the membrane that the peptides need to penetrate.
Yeah. So with my research, we are looking at targeting a class of bacteria known as the gram-negative bacteria. So it is a group of bacteria that is known to be incredibly hard to kill because they have an outer membrane that is protecting the cell and preventing molecules from entering and killing the bacteria. So so in basically look at using all sorts of molecules, and we chance upon this way in which our body naturally defends itself against infections, and that is by producing some kind of small peptide molecules. And these peptide molecules are capable of breaking through bacterial membranes and killing the cells. So taking inspiration from that, we sort of worked based on that model, and we tried to improve on that, and that's because a lot of these peptides, they are also quite toxic to the body. So we actually use chemistry skills and our knowledge in polymer engineering to basically improve on these peptides, to basically find a way to manufacture them on a larger scale, and to basically make them more selective in killing bacteria while remaining relatively non-toxic to the healthy cells in the body. And so I think we've managed to accomplish that in this research.
And the molecule has a unique architecture.
Yes. So I think so a lot of people are quite fascinated by how we actually call our polymer, and they're asking me, so you call your polymer star-shaped peptide polymer? Is it really star shape? It is actually a star shape, and the whole polymer is really small. It's about 10 nanometers, so that's really, really small, even in comparison to a bacterial cell. And so we build it in such a way that we sort of grow from a core, so that it has like arms radiating from the core, and that's how we got a star shape. And we believe that, well, we had lots of indications to show that the star shape actually sort of improves the activity of the construct itself, and that is by making it incredibly harmful to bacterial cells. and I think that the star shape really plays an important role in giving us the results that we are seeing right now.
And you've tested this theory on mice so far, I believe.
Yes.
What is the next step?
So, the next step, yeah. So, up to this stage, we have sort of obtained quite a very important piece of results, because we have managed to infect mice with a superbug infection, and then we tried to treat it with a normal sort of antibiotic, and that didn't work. I think, if I'm not wrong, half of the population of the mice actually died. But when we injected the mice with my polymer, we actually had 100% survival in the mice, and all of them were safe. So I think moving on from here, we definitely need to look at whether we can use the polymers to treat different kinds of infections. So currently we only tested on one type of superbug infection. So we're looking at if we can expand that and possibly treat other kinds of bacterial infections. And I think another very important point is we need to really determine if there are any toxic side effects of these polymers to the body. So currently we have performed basic tests to show that it is relatively non-toxic to the body. But we definitely need more in-depth assessments to prove that point. And also another thing.
Sorry. Yeah. Because polymers are particularly toxic to mammals.
Sorry,
because they are particularly toxic to mammals.
I think it depends. I think so. A lot of people have sort of a bad stigma attached to polymers or nanoparticles. But I think it with chemistry and with the advances that we have managed to achieve in the polymer field, it really depends on how you design your polymers, and you can design them to be really toxic to the body, or you could also try to manipulate the design to make them less toxic to the body. So there are different ways you can optimize that with the current technology.
So take us through a normal day for you. I mean, working with superbugs is a pretty scary thing to do. So you arrive at the office and the plan is to work with superbugs that day, sort of physically work with them. What do you do to prepare for that?
Yeah, so I think it actually sounds kind of scary when you say it like that. But for me, it was actually a slow progression. So I started off my research with just the normal bacteria strains. So and then I slowly transitioned towards working with superbugs when my skills, my experimentation skills got more developed. And every day, basically we just I reach my office and I get into the lab, and so it's just a matter of trying to protect ourselves with it's just a usual standard protocols, wearing lab coats, gowns, gloves and everything, and then we try to be as careful as possible. And basically that means not touching your face after dealing with these bacteria. And I think that currently I've worked on this for many years, so I don't really see that as much of a problem anymore.
What about funding for a project like this? The worldwide benefits are potentially immense. How do you put together funding?
Yeah. So I think I'm quite grateful because my research group has gotten funding for this particular project that we're working on, and so I think that we also managed to get funding because a few years ago I managed to get quite substantial results for this research, and so that sort of got us a competitive edge when we were applying for funding for research in the antimicrobial field. But I think in many countries, while antimicrobial resistance is sort of recognized as a very severe issue, it is still not regarded as a prioritized research area. And so what happens is that when we apply for funding, we still need to compete on equal grounds with just research in all different areas. So I think that currently there's sort of a movement to sort of try to get research in this particular area into one of the top research agendas in Australia. So I'm not sure how is it like in other countries. And by doing that, hopefully we can get the research council in our country to be more interested and devoted in actually channeling a significant portion of funding into this this field, and that will hopefully drive more people into working in this area.
Could you, for the audience, describe why the negative as opposed to the positive side of this issue is more important? I'm talking about the MDRs.
Okay. So I think that currently, so to put it on a very simple level, the issue of MDR or multi-drug resistance, probably 10 years ago, when you get a cut on your finger, and then maybe you get infected by some sort of infection because of your wound, it's just nothing, because you can just apply some cream, antimicrobial cream, and that will just solve your problem. But now with the issue of MDR, what it means is that such just a cut on your skin or wound on your body could potentially be fatal. And that's because what we're seeing now is that a lot of the antibiotics which used to work really well last time, they are no longer working anymore. And so this is just how severe the problem is. And I had lots of I actually know of lots of people where they are admitted to the hospital because of just a simple just a non-critical disease, but they actually got infected by a superbug in say the ICU, and they just pass away because of that. And so I think that it's an issue that's impacting everyone on a global scale. Yeah.
And you're working with the gram-resistant negative superbug, aren't you? As opposed to the positive, which has been seen in hospitals. Sorry, I didn't realize you're talking about the gram positive and negative bacteria. So they come from either gram positive or gram negative family. But what's I guess scary about the gram negative superbugs is that they are the kind of superbugs that are responsible for lots of infections that spread in hospitals. So that was basically what I was talking about just now. Those. So many of them are actually caused by the gram-negative superbugs. But had the gram-positive bugs been more prevalent up until fairly recently? That it's the negative which is becoming the greatest threat and needs the most funding and the most concentration of work?
Yeah. So traditionally, the gram-positive superbugs are sort of perceived as a greater threat, and that's I think also because of there's a particular gram-positive superbug that's really well known. It's called the MRSA, and So I think a few years ago, there was sort of reported all over the media as the superbug that is causing lots of deaths and everything. So I think that is the superbug that everyone knows about. But the thing is that gram-negative superbugs bugs were not really given a lot of attention, and I think over the years it has slowly crept up to be a more critical issue.
So when did you decide to work on this project? How did this all come about?
Yeah. I was very fortunate because during I think the third year of my undergraduate degree, that was probably five or 6 years ago. I actually, I started thinking, what I wanted to do, for my future career, and then I thought maybe research would be something that would interest me, and so I wanted to give research a go, and so I applied for a sort of a short-term research experience, and I got selected for the program to actually work in my current lab. So my research at that time was really focused on chemistry, and so I was really focused on making polymers, and I actually started making star polymers at that time, but it was a different kind, and we wanted to potentially use those polymers for some sort of medical application, but we didn't really know what kind of application we would be working on. And then, fast forward 2 years after that, I decided to do my PhD. And so I decided that I wanted to work on, star polymers. And then I decided I really have to pinpoint an application for my star polymers, because what's the point of, making lots of material, really find a use for it. And so, and at that time, we got into contact with one of my current supervisors. So his name is Neil. So he's Neil O'Brien Simpson, and he comes from the Melbourne Dental School. So his expertise lies on making just simple peptides to potentially kill bacteria. So we saw some sort of a potential collaboration going on there, and then we decided to work with Neil. And from then on, my research developed from there. And I think, 6 months into my research on star polymers, I basically got a very good indication that they actually worked really well in killing bacteria. And then I decided to build from there, and that's, so basically, 3.5 years of research, and that got me to this stage right now.
If you could have any help on this project, whether it is political, scientific, funding, commercial from potentially big drug companies, what do you need most to take you to the next stage?
I think currently we definitely need more funding, especially for my current research, because there's still a lot of work to be done. So although I think it's quite a significant breakthrough, but what we are at right now, the stage that we are at right now, is still considered quite preliminary, because we really need to do quite substantial work to actually bring these polymers to clinical trials. And since my paper got published, I spoke to a lot of people, and then I realized that how fast you can bring up a sort of a actually depends largely on the kind of funding, the kind of resources that you get. And if you have lots of funding from certain investors, then that would provide you with more resources to shorten that process, basically. So I think, so funding is definitely a very important thing that we need right now.
What would you use, if you could be specific, what would you use those resources for on a daily basis to make your job easier and to fast track this into being used by people?
Yes, I think for the next stage of work that we need to do, we definitely need more people working on it. So I think funding would be really beneficial to hire more postdocs or students or research assistants to help to work on this project, because so far this project has been my main PhD, is that I'm sort of the main person working on it, and so I only have a pair of hands, and that's the maximum that I can do. So obviously I get help from all sorts of people, but they also have their own projects to work on. So I think that with more funding, we can get more people specifically for this project. And with more people, you can generate more data within a shorter amount of time. And that would definitely be very, very helpful to push this project forward.
Obviously, funding always has strings attached. There are motivations behind funding projects like this. In an ideal world, which organizations would you like to receive funding from? If you could name a few, provide some examples.
Yeah. So I think this is also something that I'm in the process of learning, because in the past 3.5 years I've devoted so much time on just research, and so at this stage I'm really grateful because I've been speaking to lots of people, and I've been learning on pharmaceutical development. So I think that there are two ways that I'm pretty sure there are more ways, but what I know of, there are two ways that we can go forward. So we could get funding or investment from really big pharmaceutical companies.
Are there any ones in particular?
If you get them interested in this, so maybe like GSK, GlaxoSmithKline, or Roche. And if they are interested in taking on this product, and with the amount of funding that they have available, the future of this could be endless. And but I think there's also another approach that I guess has been proposed recently, which is basically getting small investors backing this project, and then creating a sort of a university affiliated spin-off company, and that would, I guess, also ensure our participation in the development of this drug. And I guess maybe that in some ways would also ensure sort of more control over how we want to direct our future research for this particular drug. So I think that there are multiple ways that we can go forward. And currently we're just really open to all opportunities, and we're still talking to lots of different people about this.
Could you explain what the open access movement is and the importance of that within science, and whether you would like or are currently working within that framework on this project?
I think that this open access thing has existed for quite a number of years. So traditionally, science is sort of, so for example, for scientific publications, so a lot of scientific publications are actually not open access, and that basically means that, you or your institution would basically need to pay in order to get access to those results. And honestly, like for my research, the journal that we publish in, Nature Microbiology, it's also those kind of it's also not open access. But recently, there has been sort of a movement towards open access science published in open access journals, and that basically would provide access to lots of different kinds of information to, say, the public, which means that anyone can read any journal that they like at any time. And I think that this would be really beneficial, I think, to promote better engagement between the public and scientists, because I think that traditionally scientists are sort of viewed as a sort of a close community, and sort of really distant from the public, and people don't really know what scientists are doing And even though many great research achievements actually came out of their research. And I think that open access policy actually helps to promote that sort of relationship between the public and scientists. But I think another very antimicrobial research itself. And I think a group based in the University of Queensland in Australia, they have been working on open access antimicrobial drug discovery. And so basically what they're trying to do is they're trying to crowdsource molecules from everyone, from different parts of the world. And they're willing to offer their services to basically help people to test their molecules against bacteria. And the reason for this this sort of open access community is because previously drug discovery has been sort of a lot of the preliminary stages have been done behind closed doors. So we don't sort of disclose our results until after publication or after we patent our molecule. And there are different sort of groups of people working on materials or molecules for different purposes, but you never know. Maybe one of these molecules could be actually a potential antimicrobial drug. And the reason why we don't know is because no one offers to test the molecule for us. And so what this group they are offering is that they are willing to test anyone's molecules for free, and that would probably help to foster sort of a greater collaboration across researchers, and maybe that would increase the antimicrobial drug pipeline, which is really running dry at the moment.
That is terribly exciting, and presumably
it is.
That will get the ball rolling as far as worldwide collaboration on this, because as we said at the beginning of this conversation, the entire population of the world will benefit from this, potentially.
Yes. So I'm really excited when I hear about their project. And they have been receiving quite a lot of publicity for that recently, and I'm really happy for that. And who knows, in the future I'm excited to be looking at possibly collaborating with them and seeing how we can synergize our research together.
Are there any other scientists across the world, whether it's groups of scientists, specific universities, or individuals that you would really like to work with on this project?
Yeah. So I've been quite actively following certain scientists in this area, and there's one particular scientist that got me really interested in his work. So his name is James Collins from MIT, Harvard, and Boston University. So his approach is from a more, his approach in antimicrobial research is from a more fundamental sort of angle. So where he's trying to understand how antibiotics or antimicrobial agents work. And by understanding that, the information is really useful because they could help us to design better and more effective ways of targeting and killing bacteria. So I think that there's potential sort of collaboration that between my research and his research, because I'm coming from a more applied research sort of angle, whereas he's coming from a more fundamental angle. And I think that by working with him, we could gain more understanding, for example, into how polymers actually work. So currently we have sort of a basic understanding, but we definitely need to do more work, and we also need expertise from people like him to help us understand better. And with better understanding, I always think that we will be able to optimize and develop better drugs from there.
Do you come from a family of scientists?
No, actually. So I think I'm actually the first person in my family. I mean, even my extended family, because my extended family is really big. My dad had 10 siblings. So, but I'm the first person to actually do a PhD, and I'm also the first person to work in science for that long. And well, my dad was actually a doctor, so I think that's the closest sort of line of work to mine. And I think that throughout my PhD, because I think because of his sort of a medical background, so he had some sort of understanding in the research that I'm doing. So I really enjoyed, discussing my research findings with him. And I think that, because I think research is sort of a really long journey, and there were lots of ups and downs, lots of failures that I faced. But I think that his support really gave me the encouragement and the motivation to push on.
Can you remember a point in the past when you caught the science bug? There was a spark and you decided, I want to be a scientist.
Yeah. So I think since young, I always got asked, what I wanted to do in the future, and I actually didn't know the answer. And I always said I wanted to be a doctor, and that's just because it was a second answer. Because my dad was a doctor, and it's sort of just natural for me to just want to be a doctor as well. But I think I've always been interested in science, but I didn't. But the thought of actually becoming a scientist or a research scientist didn't really come across to my mind, because it was always perceived as a very foreign concept. I didn't really know what scientists do, and it seemed like really a really difficult line of work. But when I got to my high school, I realized that I was really interested in chemistry and mathematics, and I'm really interested in applying fundamental knowledge to solve actual problems faced in everyday life. And that's why I did a undergraduate degree in chemical engineering. So that's sort of a merge between chemistry and mathematics. And halfway into my degree, I realized I didn't want to be an engineer. And that got me thinking, why am I even doing an engineering degree? And then that's why I started exploring, other careers, and then I chanced upon chemical engineering research, and then I thought that research is really what I wanted to do, because it is really just using your fundamental knowledge, for example, in chemistry or engineering or mathematics, and then utilizing these knowledge to actually create something, to engineer a solution to an actual problem. And so from then on, I decided I really wanted to do research, and I think it was at that point, yeah. So I didn't I wasn't interested in research since a very young age. But I think towards the end of my undergraduate degree, I really, I really found my passion.
Do you have siblings?
Yeah. So I have an elder sister and a younger sister. So I'm the middle child.
And are they interested in STEM?
No, actually. So one of my sisters in particular, she, I think she sort of really dislikes science. I think she sort of didn't know what she wanted to do in high school, and that's why she, stuck to science and studied, physics, chemistry, and biology. But then she realized she really didn't like it, so she ended up doing law. So she went on to, she's actually working in the finance industry at the moment. So I think that actually, come to think about it, not many people in my family are actually doing science.
And what does your mother think about all of this? She must be very proud.
Yeah, I think so she didn't really understand what I was doing previously, and I think I didn't really do a good job in trying to explain to her what I'm doing. So she sort of think that I'm just doing experiments in the lab. And I think it wasn't until my research findings got published and the research actually got lots of media attention. And with media attention, the newspapers, they basically translated my research findings in more in simpler layman's terms. And it was actually by reading those news articles about my work, she actually got a better understanding of what I'm actually doing. And I think, and she told me she's really proud, and she was asking me, why didn't you tell me you're actually working on this project last time? So I think I really did a good job in explaining my research to her.
Are you taking part in the nanotechnology conference in Melbourne in November? Are you presenting your idea there?
I'm not sure which conference you're talking about, but I think in the few months, actually, I think end of this month, there's an international biotech conference in Melbourne that I'm attending. But there's also another conference in I think sometime in November. So it's a polymer conference in Melbourne as well that I'm actually giving 2 presentations about my work.
Maybe you should invite your mum along to that.
Yes, I think I actually should.
If there was one person that you could meet in the world, whether it's within your field of work or within your field of interests, maybe hobbies, who would you like to meet?
Yeah, so I think this morning on Twitter, I was actually looking at Twitter, and I actually saw this article about Professor Yoshinori Osumi. So he recently got awarded the Nobel Prize in medicine for his research on autophagy, which is basically understanding how cells recycle their components. I saw this article, and what struck me as really interesting was that in an interview conducted with him after he got awarded the Nobel Prize, he said that so I think he's currently about 70 years old, and he said that he will actually devote the rest of his career in building a new research center for microbiology and creating an environment to really support the research work of young scientists in microbiology. So I think that really, really inspired me because, he has accomplished so much in the 50 years of his research career, and even at this age, he's really still, passionate about supporting young scientists working in my field. And so I think that he's really, one person that I really want to meet, and I'm really interested to know what he's going to do from now on. And I would really want to personally thank him for his support for young researchers.
So that was going to be my next question. What would be the question you would want to ask? So is that your question?
Yeah. So I think another question that I would be really interested to ask would be what actually kept him going throughout his 50 years of research career. And that's because from what I read online, I understood that so basically for his discovery, for his research on autophagy, he actually started this research when this research field was really not so popular, and there weren't really much done on this area. And so I think that he really had a lot of courage to actually take on research in this area, and to actually continue working in it, and eventually lead to such fantastic discovery.
What is going to happen next then? You've been working extremely hard on this paper. There are many steps along the line in terms of the project generally. You want participation, you want investment. But what about you as a human being? What do you do outside work? What are your ambitions as far as things to do?
Currently I'm actually about to finish my PhD. So I'm caught up in the stresses of trying to write out my thesis, getting my other papers published. So I think it's just the usual sort of stress faced by a typical PhD student. So I'm actually quite stuck in that at the moment. But I'm actually also at the point in time where I'm seriously considering what's the next step for me. And I think that for me personally, I'm still really interested to continue working in research, especially in this area. And I think that I'm considering different kinds of options, and ultimately what I want is to still work in a research group that would provide me with lots of support in pursuing research in this area. And regarding I think on a more personal level, I think I would want to, I guess, in the future in my career, to while actively diligently working in antimicrobial research, I would still want to I think achieve a sort of a good balance between research and personal life. I think because sometimes we get too caught up in research that we sort of neglect our health or our personal time. And I think I really want to try and avoid doing that. And I think that's one of my goals for the future.
Have you encountered any particular challenges in what you do as a woman?
There were lots of ups and downs throughout my PhD, and I think that because I'm actually still an international student in Australia, so I'm actually not an Australian. So I was originally from Malaysia, and I'm still a Malaysian citizen. So that means that the rest of my family, they are all in Malaysia, and so I think one of the challenges that I faced was that it was a bit difficult to juggle between my research here in Australia and my family back home. So last year my father passed away just suddenly, and
sorry to hear that.
Thank you. And so I was still in the process of working really hard to generate results for this research, and because of that my dad passing away, I decided to take a short break from my research and to basically return to my home country to help with dealing with all the matters related to his passing. And I think that that was a period of time where I really struggled a lot, and that's because I was still, I know I have an obligation here in Melbourne to continue with my studies, but I also know that I have an obligation to my family at home. And I decided at that point in time, family was more important for me, and then they really needed my support. So I stayed in my home country for about 2 to 3 months. So I was still working on writing my paper and doing some paperwork for my research on and off back home. But I was really focused on my family at that time. And once everything was sort of more or less dealt with at home, I then decided to come back to Australia, and it was quite a difficult transition period for me at that time as well. But I think I managed to overcome that, and then after that, I was able to devote myself back to research again.
So those are enormous personal challenges. I'm so sorry to hear about your father.
No.
Have you encountered any challenges as a woman within your field of work, or does that not enter your mind?
I think that I think in the research environment in Australia is still quite supportive of of women in science, and I think especially in my field of research, in engineering, because my research group is based in the Melbourne School of Engineering, we still have more male researchers than females. But I think that everyone's really supportive of us as females, and I actually didn't really face much problems working as a researcher, as a female researcher.
If you're speaking to girls, boys out there, sort of 12, 13 years old, what would you say to inspire them into your line of work?
I think I wouldn't necessarily try to inspire them into my line of work, because but what I would say to them is to really just to try to get more exposure, as much exposure as you can, and try to and by doing that you could possibly identify your passion, your interest in life, and that would really help to determine what sort of career that you will be in the future. And I think the reason why I would really encourage young boys and girls to do that is because when I was younger, I didn't really know much about research. I didn't really know much about science, and that's why I didn't know about I didn't manage to identify my interest in life until when I was 23 years old. So that was quite late. So I would encourage the younger generation to really open their eyes and try to experiment and try new things, and because you never know what you might find in the process.
Could you finally summarize your project and the message that you would like to send to potential collaborators, scientists out there in the world, and potential investors in your project?
Right. So I'll try to do it in a very simple terms. So basically, out from my research, so my research lies at the interface of chemistry and biology. So where I try to use my expertise in chemistry to solve problems in biology. And so out of that sort of attempt, I managed to develop a new form of a drug molecule that, based on our results currently, could kill superbugs really effectively while remaining non-toxic to healthy cells in the body. And another exciting point is that we managed to show that these drug molecules actually work in wiping out bacterial infections in mice, which means that they are really promising. And another important point is also that they actually do not cause bacteria to mutate and acquire resistance, which is a very important, a very important criteria for any form of new sort of antimicrobial drug that we develop from now on. And so what this means is that my research potentially mark the unlocking of a new treatment method for antibiotic-resistant infections. And so moving on from here, I think that my research has a lot of potential. And so we would really need lots of funding, resources to help to move this piece of research forward into clinical trials and potentially moving on and bringing it into the market. And in terms of future research collaborations, we will appreciate all interested parties from fundamental and applied research areas to get in touch with us if you have any interesting proposition, for example, in helping us to understand better how our drug molecules actually work. And so I think in summary, I think this research is really it's a platform for many interesting opportunities in the future, and I'm really looking forward to that.
Well, thank you so much for sharing your story with me and talking to me about your work. And
I'm really impressed by your ideas and this project that you're working on. Thank you.
Bye-bye.
Bye.